Texas A&M and University Corpus Christi. I've been trained as a neuroscientist, got my Ph.D. from University of Pisa in Italy, and moved to Texas in 1999 to work at the University of Texas Health Science Center. And some names will come in the presentation today. My field of work is understanding how the brain works, and that's why my presentation would be. I'm the only neuroscientist with a research lab here on campus that do cellular and molecular work. Hopefully we'll have more coming. I've been mentoring several students in different disciplines, from biology to biomedical sciences and marine biology. And my funds come primarily from the National Science Foundation and the National Institute of Health. And I don't know what else. I think most of what I do will come out of the presentation. And I'm in Tidal Hall for those who want to see the snails, the animals that I work. We can give tours. We used to do a lot of that before the pandemic. We used to have kids that do twice, 3 times a year. I think we had about 600 students over 5-6 years. Pandemic obviously changed the picture a little bit. And I'm very happy to be here.
That's great, thank you. My name is Lisa Lewis. I'm Director of User and Research Services here at Bell Library, and joined by my colleague Alexa Height, who is going to tell us a little bit about the Outstanding Faculty Speaker Award Series. So Alexa, I'll hand it over to you, and then Dr. Mozzachiodi, just jump on in when Alexa is done. Thank you.
Good morning, everyone. It's so good to be with some folks today on this chilly Friday afternoon. My name is Alexa Height. I am the Scholarly Communication and Copyright Librarian, as well as the liaison to the College of Science and Engineering at Mary and Jeff Bell Library. And we'd like to thank all of you for joining us today to celebrate our Fall 2021 Outstanding Faculty Speaker, Dr. Riccardo Mozzachiodi. The Outstanding Faculty Speaker Series is co-hosted by the library and the Center for Faculty Excellence. The selection process is competitive, and our speakers are chosen by the members of the Center for Faculty Excellence Committee. So in the fall semesters, we celebrate faculty participating in research and scholarship, and in spring semesters, we focus on faculty who are outstanding for their creative endeavors. Through this program, we highlight the work of our faculty and share scholarship across the Island University. So now I will hand things back off to Dr. Mozzachiodi. Thank you.
Hello, everybody. Let me share my screen with you. Can you see the screen? You can see the screen, right? Okay. So, and also let me close this panel over here and move it mode. Hello, everybody. So thank you for inviting me. Thank you for the Bell Library and for the Center for Faculty Excellence for selecting me as your speaker for today. Before I start the presentation, I would like to tell you a little bit more of what my field of work is. I'm a neuroscientist, but it's a very, very broad definition. So what I'm in a behavioral neurophysiologist. What it means is that I study the relationship between neurons, brain cells, and behavior, and try to understand how neurons control behaviors and the modification in behavior. And in this regard, the presentation that I'm giving you today is to deal with how we make simple decisions, and in particularly in the context of 2 forms of experience that can modify our decisions: reward and fear. And I'd like you to take you through a small journey going from behavior to cellular down to molecules. So first of all, I don't need to spend too much time to tell you about how decisions are made. I think probably everybody understands it. We make decisions all the time. And regardless who we are, if in the animal kingdom, decisions are critical for us to survive. And because to survive our ever-changing environment, we need to make decisions. But what is also very important is that we need to remember the decision we make. And this is, I stress this out because most of my work actually doesn't focus on how we make the decision as much as how we store the information of a decision that we made. And decisions also have to be flexible. What that means is that we need to be able to change them in a changing environment. And two factors that are known to modulate decisions are rewards and fear. And this is the context in which my presentation will work. So very briefly, what I would like to share with you today is the possibility to compare and contrast the way a very simple decision is modulated by rewards and fear. So what I need to do first, I need to select for you a behavior that is highly controlled by decisions. My choice will be also using a behavior that is controlled by a nervous system part that I know. Second one, even go to the single cell level, identify neurons that are capable of decision making. And finally, show you how experience changed the decision and changed the neuron activity. But first of all, I need to tell you in which organism my work functions. So obviously, when it comes to decision, everybody jumps immediately to try to study it in humans, because that's who we are. And of course, in terms of mental health and other aspects, we really want to understand how decisions are modulated in our brain. But there are some challenges. And the first challenge is that this picture shows you a brain, is that our brain is made of many, many, many neurons. The estimate says about 100 billion. And they're very, very tiny cells, about 4 microns in diameter. So studying decisions in a very complex brain with the addition of very complex decisions makes very difficult to approach the decision-making study in the human brain. But so that's why we find a way around, if you want. First of all, because decisions are universal, as I said, and they are highly conserved, why don't we study them in simple organisms, in organisms that have a much simpler brain than ours? And before I reveal the model that I work with, I want to bring you this individual here. His name was August Krogh. He was a scientist who received the Nobel Prize in 1920 for his study of the capillary system of the circulation. But his work became famous across discipline because of what Professor Krogh said. He said that for such a large number of problems, there will be some animals of choice or a few such animals on which it can be mostly convenient study. So what that sentence says is that we can find an organism that can help us answering our questions. And in this regard, this is the animal that I use in my laboratory. It's a marine snail. Its name is Aplysia californica. It comes from the Pacific Ocean. Very exotic to say. In reality, they are collected in front of Los Angeles and they're shipped to us via FedEx every few weeks. And actually, this is a picture of how we store them in the laboratory. So why do we use a snail in our research? Well, first of all, important disclaimer, I'm not the first one who used this animal to study memory and to study a decision and to study the brain. It's been used as a model system in neurobiology for about 65 years. For my particular type of work, of course, because Aplysia makes decisions. It also expresses simple measurable behaviors. It's also slow. So the behavior that I will deal with you today, there can be measured in the order of seconds. So you can quantify them very easily. A very important point for my work is that the nervous system of Aplysia has a very small number of cells compared to ours. Instead of billions, only 20,000. As you will see in my presentation, I don't even have to worry about 20,000. It's a much smaller number. A point that I highlighted here in the slide is that even if I study a snail, the function of the brain cells is extremely similar to ours. So it's almost freaky if you want. The way neurons function at the cellular level is no different between a human and a snail. Obviously, we are different than snails because we have a larger number of cells, so we can do more. A very other important point that helps my work is that the neurons are large. So if you see the photograph here on the right side, this has been taken with a microscope the students have in their research and teaching labs. And everywhere you see a little ball here in the picture, these are brain cells. And in the picture here, the brain cells have a diameter of about 40, 50, 100 microns. So they're really large compared to ours. And this is very important because we can actually study them at the cellular level. And finally, which is really important for my work, is that I can have undergraduate students leading experiments. Aplysia is a very user-friendly model for undergraduate to work. I have graduate students in my lab, but I also employ a lot of undergraduates. So when it comes to choose a behavior that is controlled by decision, it's not very difficult to choose feeding. We all have to eat. And we know very well from what we do every day that we decide whether or not to eat. In a couple of days is Thanksgiving. Thanksgiving afternoon, probably none of us is interested in the evening to eat again. Vice versa, if you're starving, you really need to seek food. So let me show you how Aplysia eats. So first of all, Aplysia uses seaweed as food. It's the Pacific nori that you find in California rolls. So I want you to focus on the right side. This is the mouth of the animal. And the structure you see in the middle here, it's called the radula. It's a grasper. It's the tongue of the animal. And what Aplysia does, it protracts the radula open, grabs the food, and brings the food inside the mouth. And every time it does this, it's a cycle that we call a bite. So through multiple bites, Aplysia can bring the food inside the cavity. So in my lab, we do not test the feeding capability of Aplysia using physical food. We use actually a seaweed extract. So bottom line, Aplysia doesn't really see very well. The way he seeks for food is using olfaction. He smells the presence of food and then starts biting, hoping that the bite can bring some food inside the mouth. So what we do in the lab, we put Aplysia in a seaweed extract. Aplysia is very excited, and we're going to start making bites, deciding to eat. And I want to show you a brief movie that a student of mine, Robert Mueller, made, which is fantastic. Look at this picture. So you see Aplysia open the mouth, you see the radula coming out and retracting in a closed position. And you count one bite. Now it's another one is coming, opening, closing, and retracting. There's no food, but imagine it that it would be food, Aplysia will bring it in. And there's a third one here. So you see, you can also count. We have a clicker, and you can count bites very easily. So again, why we choose feeding? Well, it's critical for the animal survival. It's easy to measure, and it can be modulated by many external and internal factors. So the next step in my work that I need to introduce you is that, remember, the goal of the presentation is that I want to show you how the decision is modulated by experience. And so I need to introduce you some learning aspects. Now, organisms learn a lot, but the world of learning and memory is quite complicated. If you see this picture, you see many, many branches, and you don't have to worry too much about everything in the picture, except the fact that Aplysia, a snail, shares a lot of forms of memory with humans. And in particular, the forms of memory that Aplysia shares are in this box in the lower corner. And I will show you some of these forms of memory that we used in the previous year to challenge Aplysia. So what I want to show you today is that I want to challenge the decision to eat using rewards and fear. So in this cartoon, I decided to give you an idea what could be a carrot as a reward for Aplysia, or on the other hand, a stick, bottom line, a threat. Well, in the world of Aplysia, the reward is seaweed. Again, Aplysia loves the seaweed. They never say no if you present seaweed. So this is a good reward for the animal. And as a potential threat, well, could be a predator, and like a lobster that Aplysia can encounter in the ocean. What I want to say is that in the presentation here and most of my work, we really want to see what happens, not necessarily when Aplysia makes the decision, but for how long Aplysia keeps that decision of whether or not to eat. So in the presentation here, I will show you how Aplysia can actually store information for a prolonged period of time. in particularly the time point that I will use is 24 hours because it's an indication of what we call long-term memory. So as an idea, as an hypothesis, I would think that if you provide Aplysia with rewards, Aplysia would increase the decision to eat. And on the other hand, if you present Aplysia with a threat, the decision to eat would decrease. So then we need to see if that is the case. So I will choose in this presentation two forms of experience to challenge the decision to eat. The first one is called operant conditioning, and I'll show you in a moment. The second one is fear learning. You see, operant conditioning will represent the reward, a good thing. The fear learning will represent a bad day in the life of Aplysia. So let me introduce operant conditioning. It's a form of learning that it's quite common. We all have it. We may not realizing that we use it, but through operant conditioning, an organism learns the consequences of our own behavior. What it means in very simple terms is that if some sort of a reinforcement happens in coincident, contingent with a specific behavior we make, in this case, a generalized behavior 2, that behavior 2 in the future may change its expression. It may increase or it may decrease depending on the nature of the reinforcement. For the presentation today, in particularly, I want to use food reward. So I want to give the animal rewards. And the idea behind this hypothesis is that if I present food reward when Aplysia is biting, Aplysia make an association and it will bite more in the future. So let's project this in the life of Aplysia. So Aplysia, it's a snail, it's in an environment, it can engage in many different behaviors. As I said before, Aplysia doesn't really use eyes to locate the presence of food. It smells it. So if there is the presence of food by smell, what Aplysia is going to start to do is waving the neck and start biting around until food is ingested. So the idea is that if Aplysia makes a bite and the bite is rewarded by the presence of food, Aplysia make a connection. First of all, thinks there's more food around, so it will start biting again. But what is very important, very likely, if tomorrow Aplysia comes in an environment and when it smells the presence of food, it will make more bites because it thinks that there's food around. A few years ago, when I was at the University of Texas Health Science Center, a colleague of mine, Bjorn Brems, now in Germany, developed a really great protocol to test this hypothesis of food reward learning. What he decided to do, he was putting Aplysia in a seaweed extract. The Aplysia make bites, but when a bite happening, Dr. Brems was applying not food as a reward, but a tiny electrical stimulus represented by this red lightning bolt here, happening exactly when Aplysia was biting. This is some sort of a virtual reward, if you want. If any of you saw the movie The Matrix, the very first Matrix in 1999, one of the characters in the movie says that I can pretend that you're eating a steak by stimulating a part of your brain so that you can think you're eating a steak. And that's exactly the principle behind this. Every time Aplysia bites, we give it a very tiny stimulus that conveys the presence of food. So what Dr. Brems did, he conducted 2 training protocols. In the first one, as you can see, every time Aplysia bites with the black box, the black circle, excuse me, he was applying a reward. But he also had to do some controls. And in the control he used was called yoke training. In yoke training, every time there's a bite, he was not rewarded. So the reward didn't come contingently, but the animal was presented with the same number of rewards. And then he was testing the bites 24 hours later. And you can see from the experiment on the right side for the statistics, in the reward group, the number of bites was higher. So when 24 hours Aplysia was put in the same context of the seaweed, it thought that there's food and biting more. So the decision of biting increased. Now, when I moved here to Corpus Christi, I wanted to challenge Aplysia in a slightly different way. Instead of giving it a good reward, I wanted to scare it. And so that's why we developed what we call a fear learning experience program. So first of all, how do we scare Aplysia? Well, in the real world, it would be a predator. In the lab, having a lobster and Aplysia cooperating in an experiment, it's not exactly exactly simple. So that's why we turn to a slightly different approach, using very strong electrical stimuli as a virtual threat, if you want. Now, you see that now there's a lightning bolt here, which is yellow. So every time in the presentation you see this yellow lightning bolt, it means strong electrical stimuli, in particularly 10 seconds with 10 shocks applied to the body of the animal. I want to make a point that these shocks do not hurt the animal. They do not injure the animal. They just scare Aplysia. And let me show you a movie that Robert again made, in which you're going to see a probe coming, stimulating the body of Aplysia, and Aplysia start releasing ink. Aplysia is like an octopus. It releases ink. And what the goal here is that Aplysia, by releasing ink, it will try to create confusion in the predator so that it can buy some time to escape. So let's see if this protocol works in terms of changing the decision to eat. So we pre-tested the animals, then we apply an aversive training protocol consisting of 4 trials of aversive stimuli, and then 24 hours we test the biting again. And as you can see in the statistics, the result is now the opposite you saw before. The decision to bite is decreased. So Aplysia, and it makes sense if you want, if you are under threat, even for a prolonged period of time, you do not want to have a meal. You actually focus on trying to protect yourself. So as a first summary point in this presentation, I want to show you that the original hypothesis makes sense. When we apply a reward learning protocol, the decision to eat increases. When we scare Aplysia, the decision decreases. And keep in mind, Aplysia remembers this for 24 hours, at least for 24 hours. So now I want to take you one more layer down. Most of my work happens not only at the behavioral level, but at the cellular level. And I can do this because the nervous system structure and physiology in Aplysia is extremely well known. And the feeding circuit is also very well known. So this is a photograph of a portion of the brain of Aplysia. First of all, Aplysia has a nervous system a little bit different than ours. It doesn't have a spinal cord. It doesn't have a cerebrum. It has what we call ganglia. Ganglia are bags of cells. And the portion of the nervous system you see in the photograph here is where the feeding behavior is generated. And we can take that portion of the brain out of an animal, and we can actually store it for almost several days. And the brain remains alive. And you see that there's even a map of the brain. People bother to characterize the different cells so that from day to day, I can actually go and study a different cell. And on the right side, you see a simple model of how a bite works. Remember, protraction, closure, and retraction of the radula. Well, we know quite a bit about the neurons responsible of that. And today I particularly want you to focus on a neuron called B51. Again, the number is arbitrary. Somebody gave it a name. You can give it another name if you want. So in this picture, we marked 51 in a ganglion. And now 51 is really a really cool cell. I've been studying 51 for many years because it has decision-making capabilities at the cellular level. So it's truly a decision maker responsible for the decision to bite. Well, how can we do this? Well, many years of work demonstrated this. What we do in the lab, we can take the nervous system of Aplysia, we can put it in a petri dish, and even if there's no mouth anymore, there's no animal anymore, we can still see the presence of bites. We can record from specific nerves. I'm going a little bit into physiology, and I promise that I try to be as simple as possible. But we can actually record the activity of the different systems controlling protraction, retraction, and closure of the radula, even if the radula is not there anymore. And if you look at the black boxes here, one for protraction, one for closure, and for retraction, these are recording from nerves, and you can reconstruct a bite without the animal. We call it an in vitro bite. There's protraction, and there's the retraction with the radula retracting closed. 51 is actually correlating its activity exactly when a bite happens. But there's more. As I said, B51 not only correlates, but it's a decision maker. It defines the presence of a bite. So the work of a phenomenal scientist named Romeo Nargio in the 90s demonstrated that B51 is actually necessary and sufficient for the decision to bite. So in the left side here of the picture, you see the sufficiency, because if we artificially activate 51, a bite is made. Vice versa, if we artificially silence 51, we prevent 51 to be active, you don't get any bites. So this tells you that 51 is really a critical cell for us to study. And I want to show you a little bit of what we do in the lab to interrogate 51, to ask the question to the cells. So we take a portion of the brain out, we put in a petri dish, and then we record the activity of B51. Now, what you see here, it's a technique used called electrophysiology. So what we do here, we injecting current into the cell. And you see I'm injecting 5 nanoamps, 6 nanoamps, 7 nanoamps so far. And you see that between 5 and 9, there's not much to happen. But I want to point out the difference when we inject 10. In this case, you see that now 51 comes alive. And when 51 expresses this pattern of activity, the decision is made. So this is the cell telling the nervous system, make a bite. So there's a very powerful transition between 9 and 10 nanoamps. So I spent a few minutes because some of this concept will become important in the rest of the presentation. So this value of 10 nanoamps is very important for our experiment. We call it the burst threshold. And it's a threshold, as you see, because before that point, the cell is not active. At 10 nanoamps in this preparation, B51 becomes active. So it's really the threshold for the decision, if you want. And it's an indication of what we call the neuronal excitability of the cell, how active the cell is. So just to give you a sense, if you will encounter in the rest of the presentation a high threshold, that of course means that 51 struggles to make a decision, doesn't want to become active. Vice versa, if you find a low threshold of activation, means that it's easy to activate the cell to make a decision. So this introduction is very important for me because it tells you that I can, in my lab, transition from study behaviors to study things at the cellular level. So instead of interrogating the animal and ask the animal, do you want to make a bite or not? Investigating the decision to make a bite, I can ask the nervous system at the cellular level if you want to make a bite. So the interrogations shift from the behavior to the cellular level. So now let's go back to the forms of learning that I told you before, because the point here is, if reward learning and fear learning change the decision to bite, do they change the decision at the cellular level? Do they act on neuron 51? Let's start with reward learning. So the work conducted by my colleague Bjorn Brands and by me at the University of Texas Health Science Center several years ago demonstrated that if we train Aplysia with the reward protocol, and 24 hours later we remove the nervous system and we interrogate the cell, if you look at panel C, you see that in animals that were rewarded with food, or virtual food if you allow, the threshold for activation is much lower than the animals that receive yolk control. You see, you're dealing with 6 versus 14 nanoamps. Bottom line, it's much easier to activate 51 after reward learning than in the control. And the summary data show exactly this. In the reward preparation, the burst threshold is much lower than the yolk preparation, which tells you that B51 makes more easily the decision to bite, which you expect following reward learning. So when we were here in Corpus Christi, we challenged the animal with the opposite situation. We wanted to scare the animal. We want to create a memory for the fear and see what happens to B51. So we train our animals with the aversive protocol that I mentioned before. We scare the animal 4 times over an hour and a half. 24 hours later, we take the nervous system out of Aplysia and we measure the burst threshold. And if you look at the sample data and the traces and the statistics, you see a completely different scenario compared to the reward learning. Now, it takes a lot of stimulation in 51 to make it active. In this example, 80 nanoamps in the trained animals versus 9 animals in the untrained animals, something that I forgot to mention. All our controls for these experiments are animals that never receive a shock. So the untrained are just simply animals that are handled but never scared. So you see a very big difference, which is opposite of what you saw before. So following aversive learning, the decision to bite decreases, and the decision maker, B51, also decrease its willingness to make a bite. So now I can revise the cartoon that I showed you before, because I can put at the center of the cartoon these very interesting critical neurons. So reward learning and fear learning manipulate the decision to bite in two completely opposite directions. And this is caused, at least in part, by this decision maker called neuron B51. When the excitability of B51 increases, for reward learning, Aplysia, this increases the decision to make a bite. On the other hand, when fear learning happens, the excitability of B51 decreases, meaning that B51 decided less to make bites. So you see that there's a very good consistency and coherence between what we see at the cellular level and what we see at the behavioral level. And truly, the neuron 51 is at the core of making this decision in two directions, depending on what kind of experience that Aplysia had. Now, in the last part of the presentation today, I want to take you one layer further and take you into the world of molecules, going down to the single molecules that could be responsible for the changes that you see in vivo. So let's start with reward learning. The work of several scientists, including me, managed to characterize the molecular steps responsible for reward learning. It's a complicated diagram. I'm not asking you to remember much of this diagram, except one thing, is that we identify a specific molecule that seems to be necessary for increasing the excitability of the neuron 51 and increasing the decision to bite following reward learning. That molecule is called cyclic AMP. It's abbreviated CAMP. Cyclic AMP stands for cyclic adenosine monophosphate. And the reason why I put the molecule here is just to show you what it is. I'm not expecting to remember, but because cyclic AMP, it's universal in life. You find this molecule in every living cell, from plants to animal cells. Cyclic AMP can be easily made and easily degraded. So it's a really fast signaling molecule. So, and it seems to play a role in the increased decision to bite following reward learning. So what we decided a few years ago was to do an interesting experiment in which we tried to ask, why don't we try to completely bypass the learning? Okay, forget about reward learning. What happens if we artificially inject that molecule called cyclic AMP inside a neuron? Can we actually convert a naive cell into a cell that learns the reward protocol? Can we truly switch the cellular activity just simply by injecting that molecule inside the cell? So we conducted this experiment in which we take naive brain this time. So we take the nervous system out of an animal that has not had an experience. We keep them in isolation for that purpose. And we record the activity of neuron 51. And then we proceeded with a tiny little syringe in injecting cyclic AMP inside the cell, inside neuron B51. Remember, B51 is large enough that I can actually stick 2 electrodes on it. So we inject like a little syringe cyclic AMP. Then we put the nervous system into an incubator. We come back 24 hours later and we test B51. We interrogate the cell again. And what you see from the statistics is that in the cells that were injected with cyclic AMP 24 hours after the injection, the threshold for activation is lower than the cells that were just injected with the vehicle in which cyclic AMP was dissolved. So actually, the experiment worked. If we artificially increase the concentration of cyclic AMP inside the cells, we were able to convert literally transform a cell that was naive into a cell that behaves like it learned reward learning. So that was quite impressive for us that that could happen. Well, a few years later, in my lab, working on fear learning, we wanted to replicate a similar experiment. My team over the years managed to characterize the signaling cascades responsible for peer learning. And again, even if you don't worry about learning what's happening here, I want to point out that there is a molecule called cyclic GMP. It's different. It's not AMP, it's GMP. That seems to be necessary to decrease the excitability of the decision maker 51 and therefore decreasing the decision to bite. So cyclic GMP is only similar in the name of cyclic NP because it's completely different in the synthesis, but similar to cyclic GMP can be easily made and easily degraded. So a few years ago in my lab, we tried to ask a similar question of the experiment I showed you a few minutes ago. What happens if we decide to completely bypass the aversive training? What happens if we bypass scaring the animal, but we just inject this molecule, cyclic GMP, inside the cell? Can we now convert a naive cell in a cell that feels the scare? Can we convert B51 from a cell that doesn't have any experience into a cell that experienced the threat? So a former undergraduate student of mine actually performed this experiment a few years ago. The experiment protocol is very similar to the one you saw. We take a naive brain, we interrogate 51 in a pretest, and then with a tiny syringe, we inject cyclic GMP into the cell. Then we put the brain into an incubator, we come back 24 hours later, and we stick an electrode again, and we interrogate B51 again. And from the statistics, you see that the results are very, very different. It's the opposite of what you saw following cyclic NP. Now, the cells that were injected with cyclic GMP, they have a threshold for activation that is way higher than the cells injected with the vehicle. That means that in this case, just simply injecting cyclic GMP converted it, a naive cell in a cell that felt the fear. So it's like we injected with a little syringe the memory of an event inside the cell. And in this particular case with cyclic GMP, the decision would be decreased because 51 would have a much higher threshold, would not be making the decision to bite. So at this point, this is really my complete summary, if you want, of the work that I show you so far. Who wants to point out that using a simple animal and a simple decision, we can actually dissect how the behavior changes, but also go at least 2 layers down, identify the neurons responsible for the behavioral changes, and how those neurons influence the behavior, and ultimately even the cascades of molecules responsible for the changes. And as a summary, reward learning, applying food rewards to Aplysia multiple times, imprints a memory to increase the decision to bite, because Aplysia obviously thinks that there's availability of food, up to 24 hours. And that increasing decision to bite is regulated, at least in part, by this neuron called B51, through an increase in the ability of the cell to making the decision. And that those changes are modulated by a specific molecule called cyclic AMP. On the other hand, if we apply a bad day in the life of Aplysia, a threatful, fearful events in the life of Aplysia will decrease the decision to bite up to 24 hours. And that decision to decrease the biting is again modulated by a neuron called B51 through a decrease in the ability of B51 to become active. And in this case, the molecular cascade is exactly the opposite of the one for the reward learning. A completely different molecule is utilized for the decrease of decision. So to complete this presentation, why do we bother to do this kind of work? Obviously, as scientists, we have a hunger, a thirst, an excitement to study how things work. But in this particular case, I want to point out that understanding how decisions are made in a very, very simple organism, which, by the way, is very similar to us in terms of cells and molecules, can be extremely helpful to understand how more complex animals, including us, can perform similar functions. And second, because if we characterize and have a picture of how decisions are made and stored in an healthy brain, we can take that knowledge and try to understand those conditions in which the process of decision making may be affected, either impaired, and we know that aging and malnutrition affect the decision to make things, but also those conditions in which decisions are made in an incorrect manner. And a great example of this is post-traumatic stress disorder. So finally, I would like to spend a minute to thank you, the people who have been working in my labs since we moved here to Corpus Christi. 34 undergraduates were trained in my laboratories, 11 graduate students, work in my lab. I cannot be more grateful to Dr. Marcy Wainwright, who is my long-term collaborator. She's my partner in crime. She's the person behind a lot of work that you saw. When she was not performing experiment on firsthand, she's also behind training the students, graduate and undergraduate, working in my lab. And I want to thank the sponsors who funded my work over the years. I want to thank the College of Science and Engineering for the continued support, the Texas Comprehensive Research Fund that funds my work multiple times, and currently two institutes at the National Institute of Health who have been funding my work, the National Institute of Neurological Disorder and Strokes, and the National Institute of General Medical Sciences. And before I complete my presentation, I would like to spend a minute to dedicate this presentation in the memory of my mentor, the person who trained me as an undergraduate, as a graduate student, Professor Marcello Brunelli, who is the gentleman here on the left side, who passed away in the fall 2020 because of COVID. Professor Brunelli taught me a lot of things I know. He also taught me, gave me the passion, instilled me the enthusiasm to go after processes and how to understand the nervous system. He was also instrumental in leading the path for my next career when I finished my graduate school and I decided to go to work to work with the gentleman on the right side. He recommended me to an old friend, and this old friend is the individual on the right side, Professor Jack Byrne, who works at the University of Texas Health Science Center. Professor Brunelli's work was fundamental in understanding how the brain works. And I pull out here an historical paper that he published in 1976 in the journal Science, in which he was the first person to demonstrate the role of cyclic MP, the molecule that I mentioned a few minutes ago, be responsible for changing neuronal activity. And that study was actually done in Aplysia, the mollusk that I show you today. The picture here on the right side was taken to celebrate this phenomenal accomplishment. This picture actually is taken at Columbia University. Professor Brunelli is the gentleman in the center. His colleague, Jack Byrne, is actually on the right side. And their mentor is in the photograph as well, and is the gentleman here on the left side. His name is Eric Andel, who went to receive the Nobel Prize for Physiology and Medicine in 2000. So I want to dedicate this work to Professor Brunelli because we try to keep the legacy of going after very simple mechanisms that can be translated in important mechanisms across species. And at this point, I want to thank you very much for your attention. I wish you a fantastic Thanksgiving holidays, and I'm happy to take any question. Thank you.
So thank you, Dr. Mozzachiodi. I was asked to give a brief introduction for you at the beginning of this presentation, and a faculty member came to my office with a budget major budget flaw. And as you and I both know, Dr. Mozzachiodi, budget comes first. So I needed to take care of that. So with Lisa's permission, I would like to give what would have been an introduction, but now what's even, I think, a little bit more meaningful after you've seen the incredible amount of work that's been done here. I'd like to tell you a little bit about Dr. Mozzachiodi, and having known him for about 15 years now, I've seen him in many different environments. The one that I see him in the most is running down the hall to, here, and from his lab. He never walks anywhere. He runs wherever he goes. He is full of energy, and that energy is felt in his classroom and in his laboratory. He briefly mentioned that he had undergraduates in his lab. He has many undergraduates in his laboratory. And he also even has had high school students in his laboratory. Can you imagine a bus full of students entering your office? This is kind of the same scenario that he had going on a couple of years ago before COVID hit, with busloads of students from a middle school here in Corpus Christi would come into his lab, and he would show them what he was doing. It's difficult for you all to understand how important this work is when you're seeing it in this two-dimensional way. But these Aplysia are actually large. They're not little snails like the ones we see in trees. They are large snails, almost the size of a kitten. And he works with these animals. There's a lot of upkeep and maintenance in doing this science. He has acquired money from the National Institutes of Health, the same organization that you've seen Dr. Fauci from. And he's done all of this in the confines of a laboratory here at our university, without a hospital behind him, without any medical, any real highly sophisticated equipment that most neuroscientists have at their disposal. He has made do with very little, and he has done so much with the amount of equipment, with his wife, who's also his lab assistant, and with the students that he has had in his laboratory. One of which, just to give you an example, just had an interview with Harvard University. The stature with which he teaches and with which he brings up his students to become real professionals in the neuroscience is so greatly appreciated by our department. And we are so thankful that more and more people are getting exposed to the important work that he's doing. Thank you for giving me the chance to speak on his behalf.
Thank you, Sherry. And actually, the fact of the student leaving from here, it's very important. The student that Sherry mentioned just began to work at Harvard Medical School. And that's the idea that you can transition with a system to the other. She worked with me on starvation, the consequences of hunger and starvation in memory formation. And now she's working on post-traumatic stress disorder. So the idea is that we can study simple organisms. She studies snails, now she studies rats, but these organisms can teach us a lot about what's happening, even if they are not as complex as us, but they are basically functioning following the same rules.
Awesome, awesome work. Thank you.
Thank you, and you can have stuffed animals. This is a student of mine who made a stuffed Aplysia.
Are there any questions for Dr. Mozzachiodi? I kind of had one. It has more to do with, like, how do you house and care for all of these little creatures that you're studying?
Well, we have aquarium systems that are quite self-sustained. We do not. Sherry mentioned a moment ago the animals are not small. We study adults. There's a big difference, of course. We are not studying animals during development. We study fully adults. There's a way for us to figure out that are adults. And so these are the two systems that we have in the lab. There are totally controlled. The water is controlled for temperature, is controlled for salinity. We make our own water. So it's like the Pacific Ocean water, except the pollutants. So instead of, actually, they're living better than the water in Los Angeles, probably. And we were very concerned about the spill, oil spill that happened few months ago. We basically control every variable. And honestly, unless there's something bad happens when the animals are in the ocean and they get sick before they come to us, they survive in very good conditions. We control everything. They eat every 3 days. We control the amount of food they do, and again, they're quite happy. Unfortunately, we cannot let them run in the aquarium alone. They have to be put in boxes because otherwise they mate, and mating mess up their hormones. And one thing that they do very quickly is that they stop biting. And so if we study the decision to eat and they refuse to bite, there's no way for us to do that. So, and also because if we let them free, that they have the possibility maybe to stick into pipes, and we don't want that to happen. But again, there are two control systems. Each of them is about $50,000, and but it works pretty well. We cannot reproduce them in-house. And there's a facility in Miami that actually grow them from eggs, to adults, and it's a really great because they can give you a stamp of approval about their age. The problem is that often they are interbred, and so because we study something that is really, we want street marked animals, so that's why they're wild caught in their environment.
Okay, I'm not seeing any questions in the chat. Last call for questions. Some people are thanking you. Incredible presentation.
Thank you.
Okay. Well, thank you so much. Really enjoyed it. I never knew that much about snails, but it's an interesting little window onto that difference between reward and the fear, sort of the fear factor there. So, and fascinating that their neurons are so much larger than ours. That's an interesting.
Yeah, that's a good advantage
detail.
for us. In terms of function, they're identical. There's a professor who wrote several books and work. He's a leader in this field, and he wrote once that he doesn't really, it's not really interested in the snail per se, just want to understand how his daughter brain works, so that he can understand what decision the daughter makes. So, and then that's summarize exactly what we do. I mean, we love the snails. We try to keep them as healthy as possible. We have a tremendous respect for the sacrifice that the animals make. Unfortunately, most of our experiments are endpoint experiments. They're sacrifices that we need to access the brain, and so we do everything to minimize whatever pain they have. But our idea is that that they can open gates for us to understand how the brain works.
That's great. Thank you, everybody, for attending today. Congratulations, Dr. Mozzachiodi.
Thank you very much.
Outstanding faculty speaker. All right.
Thank you. Bye-bye. Have a good day and a good holidays. Bye-bye.
You too.
So, Dr. Mozzachiodi, if you're still there,
Yes.
you should be, if you have not already, receiving your thank you gift from us. So, oh yes, yay!
Unfortunately,
Awesome.
I put it in a place which is transparent, so nobody could see it. I apologize for that. Sorry. Yes, it was a big surprise. Thank you so much.
That's great. Well, we really appreciate your participation and your contribution to this series. So thanks very much.
Thank you so much.
All
Bye-bye.
right. Bye.
Bye.